Okay. Thank you all for coming for today's Neuroscience Center seminar. I'm pleased to welcome Dr. Daniel Gonzales as our speaker today. So Dr. Gonzales is an engineer and neuroscientist who studies how neural computations that underlie, most recently, neuroprosthetic control. So he uses a variety of approaches, including kind of development of new techniques that allow kind of simultaneous recording, electrical recording, optical recording methods to try to understand how neural circuits kind of contribute to these behaviors in a very wide scale kind of format, certainly bigger than we do one neuron at a time, shall we say. So a little background on Dr. Gonzales. He obtained his Ph.D. in applied physics from Rice University with Dr. Jacob Robinson, and that's actually where I got to know Dr. Gonzales. I first met him in person at a worm meeting. So the worm community has these annual meetings, and there was a neurotopics meeting in Wisconsin-Madison. I happened to be his poster judge, and that's where I first got to know him in person. But before that, he's a prolific contributor to social media of all of his activities, particularly his interest in barbecue. And so that's actually how I got to know him very well. I heard you sold out recently to, your smoker is now like, yeah. So anyway, it's fine. We'll let it slide for now. After that, he did a postdoc in biomedical engineering at Purdue with Dr. Krishna Jayant, where he moved from some of his studies, which actually used C. elegans as a model system, to studying mouse brain and behaviors and so forth, which is what the work he's going to be talking about today. So since 2024, he's been assistant professor of biomedical engineering at Vanderbilt with an affiliate membership at the Vanderbilt Brain Institute. He's already have an impressive track record of productivity, having just started his position, including papers in Nature Neurotechnology and Nature Communications. He's also been successful at getting funding, including support from the NSF IGERT program, NSF GRFP, as well as a graduate student, postdoctoral enrichment from the Burroughs Wellcome Fund, and most recently as a Hannah Gray Fellow from the Howard Hughes Medical Institute. So I'm delighted for Dr. Gonzales to be here and tell us what he's been working on.
Thanks for the introduction. So it's funny, I think already in my couple of meetings, I've already had at least one person say, "Oh, I know you from Twitter." And I keep telling myself one day it's going to be like, "Oh, I read your paper that was really awesome." Not that I saw your tweets that were funny or about barbecue. We're not quite there yet. Maybe in a few more years, I'll be known more for. I'm happy to be known for barbecue also, but for tenure purposes, I would like to be known for scientific work as well. Yeah, so that was a great introduction. I covered just about everything. I'll talk about what my lab's been working on. We opened in January of 2024. So Kevin emailed me maybe almost a full year ago to give a talk, and I chose the latest date possible, and I said maybe we'll have something by then, right? Because we didn't even have, our lab didn't open until last summer, and so we're still very much getting up and running, but I'm excited to tell you about the things that we are working on. So just to kind of very broadly introduce what my lab does, we kind of work at the intersection of neuroengineering and neuroscience. So most of what I'm going to talk about today is about neural interfaces and developing and manufacturing neural interfaces. And kind of my core skill set, I would say, is in things like microfabrication, nanofabrication, making really tiny devices. And more recently, been developing how, not just developing these tools, but how do we actually translate those tools to actually study the brain, not just record from the brain, but actually use them to study neural circuits and animal behavior at a really fundamental level. And so we're going to be mostly spending our time on this side of this chart, but I very much, on the long term, have interest in using these tools to study really fundamental processes in the brain. And so just as a very, very general outline, we're going to spend most of our time talking about these flexible polymer-based neural interfaces that we design and manufacture in my lab. We primarily, technologically, what we really want to do is integrate these polymer devices with two-photon imaging, so we can both electrically and optically record from the brain, and we'll talk about why and why I think that's a good idea. And then we'll spend a little bit of time here about how we're also translating these flexible and biocompatible tools to larger brains as well, to monkey brains, not just for mice, which we'll spend most of our time on. But to kind of motivate and how do I think about our work and why do we make these tools and what are we actually trying to do and what do we want to learn about the brain? I always come back to the really huge spatial scale of the brain. This is something that I find is maybe one of the biggest challenges of neuroscience, is that the brain, you're very much familiar with, probably the brain spans many orders of magnitude in scale, right? From ion channels to subcellular to cells, all the way up to these, centimeter-sized chunks of brain, right? Everybody in here may or may not study the brain at kind of one or multiple of these scales. And so anatomically, the brain is incredibly, incredibly complex, right? And so that's one challenge. What I find even, an added challenge on top of that is the computational complexity of the brain, right? And so depending on where you decide to record and what kind of recording device you're looking at or you're using, you'll record all different types of neural dynamics across each of these scales, right? So at the subcellular scale, you can, shove an electrode into a dendrite or an axon, for example, and record spiking or, these unique neural signatures of activity at this very fine scale. You're very much familiar with probably the action potential at the cellular scale, and that's kind of being summed, all this subcellular activity being summed up. A lot of my work focuses more at like the population scale, so the activity and spiking of many of these neurons kind of working together in unison. But then there's even the larger scale, right? More like the local field potentials or EEG type recordings that give you more like oscillation type activity from hundreds or thousands or even millions of neurons, right? And so somewhere along this the scale that we have, we kind of get cognition, right? We kind of get animal behavior, all these different puzzle pieces and different types of activity and different at different anatomical cells, somehow fit together to give rise to your ability to move and behave and sense and react to the external world, right? And that's just the overarching goal and question of neuroscience, is trying to fit all these puzzle pieces together to see, like, how do we move, how do we behave, how do we think and make decisions. And so I'll give you just a really clear example of kind of looking at multiple of these scales for very, very simple behaviors, also across species. So let's just focus on motor control, right? Just moving like an arm, for example. So if you were to study this in a monkey or maybe a macaque, this is a simple center out task. So the animal just has to kind of put its finger, or sometimes uses a joystick, and a target is presented, and then it moves its arm to the target, right? And you can see the animal gets really, really good at this task. Here's a bunch of trajectories of the arm moving to one of these. Okay, sorry. I'm horrible, notoriously horrible with microphones. I like to kind of move around a little bit. So you can see that these are arms. This is just behavior, arm trajectories, right? If you look at neural dynamics, so if you plug in an electrode array and try to record from, let's say, hundreds of neurons using a really state-of-the-art neural probe, for example, we can consider this to be really high-dimensional data. This would be 200 neurons. That's a really great recording, especially in a large animal like this. And maybe you're familiar with the idea that 200 neurons is actually really difficult to analyze. Developing toolkits to understand a data set that's 200 dimensions is really challenging. So what we often do is break that down into just a few dimensions using dimensionality reduction. And what has been really consistently found in motor control is that you don't need 200 dimensions to kind of understand this behavior. But if you kind of break this 200 dimension data set down into just a handful, each of these trajectories is sort of represented on this low dimensional space. It's just a way of taking really high dimensional data and finding that we can, maybe the brain operates in really these low dimensions and sort of somehow computationally takes high dimensional data, transforms into low dimensional data, and preserves behavior in that way, right? And so this is an entire field of research by itself, is looking at neural dynamics and these what we call low dimensional manifolds. And so that's one scale. This is like more the mesoscale, right? Hundreds of neurons working together. If you zoom in a little bit, let's think about more like the circuit scale. This is still a motor behavior, very different animal, obviously in this case a mouse. All the mouse has to do in this case, it's just learning to basically press this lever. Here's a tone, and the animal just has to kind of click this lever back and forth. And it learns to do this over the course of like 5 or 10 days. And in this case, this particular paper was looking at inhibitory neurons. And so looking at inhibitory circuits, and found that inhibitory control is really, really critical for this animal to learn even this really simple behavior, right? So we've gone from hundreds of neurons to a handful of inhibitory neurons, both really, really important for controlling a simple arm movement, right? And so now let's zoom in a little bit more to the subcellular scale. This is the exact same behavior. This is actually the same group. I take a lot of inspiration. This is Takaki Komiyama's group at, I believe, UCSF, maybe UC San Diego. They train animals to do this exact same behavior, but in this case, instead of looking at individual cells, in this case inhibitory neurons, they're looking at pyramidal neurons and imaging the dendrites here. And maybe you're familiar with dendritic plasticity. This is a dendrite. Each of these little bumps here is a dendritic spine, and these spines are incredibly dynamic. They grow and they grow and they shrink, and they can even completely disappear. Whenever we talk about the brain as plastic or the brain can rewire itself, it's these dendritic spines. They can physically appear and disappear and make new physical connections with other neurons. And what they found in this case over the course of multiple sessions of this animal learning this task is that dendritic plasticity. So for example, here's 2 arrows, there's no dendritic spines. Here's 2 arrows a few days later, these spines grow and pop up, and then a few days later, one of them disappears and one of them is still there, right? And it's been continuously found over and over from many different learning paradigms that this plasticity at this subcellular scale is really important for learning and actually retaining and reproducing this behavior, right? So this is just one example of a simple arm movements across different species, how the subcellular circuit and these mesoscales are all important, right? So how do we one of the things that I look to in the long term is how do we fit all these different principles and findings together into one sort of holistic picture about how the brain works and how do we learn things and how do we generate movement and behavior. Right? And so that's, again, what I think is a major challenge of neuroscience. And so we have different tools to probe the brain at different scales, right? And so maybe you do patch clamping, God bless you if you do. I've tried it for a little bit, never got good at it, a really challenging problem. But we use patch clamping to study the brain at the cellular or subcellular scale, really challenging technique to record from individual neurons. Much more common is maybe an extracellular array to record from hundreds of neurons or dozens of neurons. Imaging we'll talk about a lot also. It's very multiple modalities from subcellular up to the hundreds of neurons or even entire brain regions. Or maybe you do like MRI or fMRI if you really need large chunks of the brain, right? Or maybe a human brain non-invasive recording, right? So we have this entire toolkit to kind of work with, and there's just a few technologies that neuroscientists kind of have at their disposal to probe the brain at each spatial scale individually, right? So we just saw in this example, they were using different tools, imaging, and electrophysiology to look at individual scales sort of at one time to see how is this anatomical scale important for this particular behavior. But what sort of in my lab, what we want to do is think about how do we combine tools to cross multiple of these scales simultaneously to, again, try to get a more holistic picture, monitor multiple of these different dynamics and anatomical scales simultaneously so we can kind of get just a better picture of what is actually happening whenever all these pieces come together to give rise to behavior. And so what we're going to focus on today is our efforts to combine electrophysiology, specifically these microelectrode arrays, with imaging to give this multi-scale recordings from the brain. And we can talk about, we'll spend just a few slides on why I think that's a good idea and what kind of platform that gives us. So let's talk about electrophysiology, right? Hopefully you're familiar with it. You take this electrode array, you shove it into the brain, and you can record activity from individual neurons and also oscillations from many neurons. So I would say the state of the art for this, maybe not like the most neurons you can get, but both very state-of-the-art and has been widely adopted by the community, is a device called NeuroPixels. So if you zoom in, this is the part that actually goes into the brain. It's a silicon probe. It's like I describe it as like a tiny toothpick or a tiny fork that goes into the brain. It has this array of recording sites, in this case almost 1,000 total. You can record from about 400 of those sites simultaneously, so you can't record from all 1,000 simultaneously, just about 400. And this is state-of-the-art. This has been really revolutionized our ability to record from the brain from a large number of cells in the mouse and also the non-human primate and even the human brain in a few cases. And gives us a lot of data to work with, right? And so here's what an example would be like from, in this case, a macaque. So you can get a huge amount of data out of the brain at one time. Each of these little blips is a spike or an extracellular action potential from an individual neuron. Here's a few of those spikes that have kind of been sorted out from the data. And this is really the power of electrophysiology. This is why electrophysiology remains kind of the gold standard for neuroscience, is because we think of these spikes as the ones and zeros of the brain, right? We think of them as the computational unit of the brain. And so electrophysiology, with electrophysiology, we can record fast enough to record these spikes that happen on a millisecond timescale, right? And so the big power and benefit to electrophysiology is the high temporal resolution to give you single spike resolution. But it also has a lot of drawbacks, right? And so if we want to, for example, here's one probe that I was putting into the brain during my postdoc. If I want to record from more neurons, I need to insert more probes, right? So that's more damage. It's really challenging to get multiple probes into a small area at one time. Another one is that electrophysiology tends to be quite biased also. So whenever we're looking at these spikes, for example, here, we're almost exclusively looking at excitatory neurons. So maybe if you have like 100 neurons you're looking at, probably like 95% of those cells are going to be excitatory cells, pyramidal neurons, a very, very small subset of your cells because of both the anatomy of the brain and also the underlying physics of these recordings. You're going to be recording from a very, very small subset of actually inhibitory neurons. And even if you can record from inhibitory neurons, you're not sure exactly what type it is. There's all these different types of inhibitory neurons that exist, right? So we're very biased in our recordings to excitatory neurons and very much less so inhibitory dynamics, which we just showed in the mouse, for example, for that simple lever press task. Those dynamics are really important for the animal's ability to learn to perform that task, for example. And then another one that's a little bit less talked about is that electrophysiology, even with the really state-of-the-art high-density probe, is actually a very sparse sampling. So here's, I just kind of threw this together. This is a electron micrograph or a 3D reconstruction of a 1 millimeter chunk of the mouse brain. I really hate these images. They're so, I mean, they're beautiful, but they're like, they give me such a sense of dread in terms of, what are we even doing? You know, we shove this thing into the brain. Look at how incredibly dense this network of cells is. And then here's a pretty good I tried to scale this silicon probe to about the right scale so you get an idea of here's the size of the probe versus the size of this little chunk of the brain. And so maybe if you have like 20 recording sites within this area, you're going to record from at best about 20 neurons. But there's about hundreds of neurons even within this really dense mesh, right? And so we're sampling a really small number of cells within this local area. And in addition to that, we're really missing this rich set of subcellular data, right? Our electrodes are very much going to be recording spikes from or action potentials from the cell body. We're going to miss or have a really hard time recording from individual axons and dendrites and all these other things that, again, are really important for contributing to learning and behavior, right? And so electrophysiology has some distinct disadvantages, right? And so in addition to electrophysiology, we have other tools at our disposal. So we talked about we're going to try to combine electrophysiology and imaging. So that was electrophysiology. What is the state of the art for imaging? I would say that is multiphoton calcium imaging. Again, we can argue multiphoton voltage imaging. There's all these other ones, but one that's been widely adopted by the community is 2-photon calcium imaging. And so in this case, we can physically look at the brain. We can get images from the brain. In this case, this is calcium imaging from excitatory cells in this case. And you can see how incredibly dense this is. This is about a 1 millimeter square. There's, I can't remember exactly, there's something about 400 or 500 cells that I was recording from in this particular session. So a very dense recording, and maybe within the same field of view, we could put 2 electrophysiology probes, for example, for at best, a few hundred cells, as opposed to, about the 500 that we're imaging from in this case. This is excitatory cells. What's nice is that this is a genetically labeled technique, right? So we can label different subsets of cells and different circuits to image and look at the activity of different things. So here's 2 different types of inhibitory neurons that you can't see in this case. And again, these inhibitory neurons are really, really hard to capture at scale with electrophysiology. So with imaging, we can target them, express calcium indicators, and watch them flicker and fluoresce as the animal is behaving or doing some type of learning task. And these are two different sets of inhibitory neurons. This is I believe this is somatostatin and VIP cells, or maybe vice versa. I forget exactly. And then what's also another really powerful aspect of imaging is that we can zoom in, right? We don't have to look at the cellular scale. We can zoom in and look at individual dendrites. We can see the activity in dendritic computations. We can even zoom in even more, and in some cases look at individual dendritic spines and the activity of individual spines. You can look at axons, right? So we can capture all the subcellular scale things that electrophysiology also misses, right? And so the power of imaging and why it's been widely adopted is that we can genetically target individual circuits and also have a very high spatial resolution for either very dense recordings or subcellular recordings, recording from very fine structures in the brain that we miss with electrophysiology. But just like electrophysiology, imaging also has disadvantages that maybe you're familiar with if you do this technique. The first one is that I talked about electrophysiology, you get, this is an electrophysiology recording from this cell. I just take us one step back. Here's a couple of cells that have been labeled with a calcium indicator. And this barely you can see in red here is a patch pipette that is also recording from that same cell. So both imaging from that cell and recording electrically from that cell. And so electrophysiology, again, the power here is that we can recapture individual action potentials from that cell. Here's 2 action potentials, 1 action potential, and 3 action potentials, right? So we can capture these ones and zeros of the brain. So really powerful, right? Because we think those ones and zeros are really important for cognition. But with imaging, maybe we can see the cell and see the cell next to it as well, but we're very much losing the single spike resolution that we have of electrophysiology, right? And so maybe there's some fancy computational techniques to kind of deconvolve, if you're familiar with deconvolving data, to get estimate how many spikes were happening at any one time in the cell. But this very clearly shows that, if you have 2 spikes, it's very hard to distinguish those spikes from these calcium traces, right? And so that's the big drawback of calcium imaging or imaging in general, is that we're losing that single spike resolution. And there's a few others that are kind of obvious. The imaging depth, right? So in my experiments, we're very much, if we're imaging from outside the brain, we're very much limited to recording from the core animal cortex. There's some fancy things you can do, like insert a big lens into the brain and image deep into the brain, but that's also really challenging. So you're very much limited in depth by imaging. And also, if you're very interested in like translating your technique, you're gonna have a hard time, genetically expressing an indicator in a human or a larger animal as well, even though it's being done a little bit in non-human primates at this point. And so, this technique is difficult to translate, right? So we definitely have some drawbacks to imaging as well, even though it's also a powerful technique. And so that's why I'm kind of trying to, spending a lot of time talking about this, is because both of these tools and techniques have really, really powerful advantages, right? So single spike resolution, high spatial modularity, genetically targeted recordings. But we typically do these either one recording technique or the other, right? And what we'd really like to do, and maybe you can envision, because we spent some time talking about it, if we can combine these tools, we can kind of get the best of both worlds. We can get the genetic targeting and spatial modularity of imaging with the high temporal resolution of electrophysiology, and have a really powerful platform for looking at the brain across these different spatial scales with single spike resolution to get, again, kind of a more holistic picture of what these neural circuits are doing during learning and behavior. The challenge, though, so let's talk about the challenge of why this isn't done very often. And so there's multiple challenges. The first one, when it comes to electrophysiology and these probes, is that number one, these devices are very opaque. They're made out of silicon, which kind of has this metallic sheen to it. You can very much see it's not transparent. It's not optically compatible, and so you can imagine that you can't image in this area at the same time, because you're going to have the light is going to be blocked by that probe, and also every time you shine light on that probe, you're going to get these really large light artifacts as well. So that's one challenge. That's one reason why it's difficult to combine both of these techniques. Another one that's a little bit less talked about, but I still think is really a huge challenge in the field, is that so whenever you're inserting a probe up close to the brain, it looks something like this. On the back end, it looks like this, right? So this is a rigid probe. So if it's a linear probe, then all the way straight back here, you have all these connectors, you have your amplifiers. On top of the animal, basically, you have all these different things that kind of impede your ability to you couldn't put an objective right here, is basically what I'm saying. You can't bring in a microscope and try to image right here in the same area. Maybe you go in a really extreme angle with the silicon probe or something crazy like that, but that's really difficult to do, right? And so just for multiple reasons, combining a traditional silicon probe with optical imaging is they're largely just incompatible with each other for all these reasons. So we have this really powerful potential for a platform, but because of certain challenges, these two techniques are really too difficult to combine. And so probably in the last 10 or 15 years or so, certain groups have started to develop new technologies that are not based on this rigid silicon material. And so people are slowly starting to turn to polymer-based neural interfaces, largely because they're more biocompatible, right? The brain is really soft. It's sort of like, I like to compare it to like Greek yogurt, right? And so if you're taking something really stiff, like a tiny miniature fork made out of silicon, you shove it into the brain, that's very soft. There's a huge mechanical mismatch there, right? And so people have tried to develop these flexible devices that are more compliant with soft neural tissue and therefore higher quality recordings, better long-term interfacing with the brain. There's a lot of reasons why you would want to use a polymer-based material. And so this is, again, been going on for decades. There's now many startups that are also using these type of devices, trying to translate them for human purposes as well. What's also really nice and been even more recent than developing these flexible probes for like the biocompatibility purposes, that's kind of the main focus of the field, is that these polymers are also naturally transparent. So here's just a couple of examples from the field. Here's an entire cranial window. The entire thing is actually covered with a probe, like what you're seeing over here, a polymer-based probe, but you can't even tell. It's completely transparent. The polymer itself is very transparent, and in this case, the electrodes are made out of something called graphene, is also very transparent. So you can't even see the recording electrodes. If you zoom, they're kind of have been highlighted here in this grid, and you can see they're imaging directly through this and doing calcium imaging of neurons right underneath that grid that's sitting on the brain surface. Here's another one. It's slightly less fancy. It's the same polymer in this case, but the electrodes are made out of platinum, and so that's why you can actually see the electrodes in this case. It's a metallic, so it's not transparent, but this still overall transparent enough to, again, image directly through that, in this case of blood vessels and vasculature. This is where my postdoc work was also in this field, and so this is something that I worked on while I was at Purdue. Again, it's very similar to those other probes that we just saw. So this is a flexible polymer-based electrode array that you could lay on the surface of the brain. And then again, because this is flexible, we can kind of move all the connectors and all those things that get in the way of the objective. We can put them off to the side, and we can have our objective right over the top of the animal to actually image over that craniotomy at the same time. And so here, that's what you're seeing over here. The contrast of this projector is a little bit off, but there's a ton of, all this, the green channel here is again, probably around 400 cells that we're imaging directly underneath that electrode array. So we can look at both electrophysiology on the brain's surface and calcium activity that's happening directly underneath that grid. And so here's the main focus of my work was to record from a part of the brain that senses touch, specifically whisker touch in animals. And so we would basically flick a whisker here and then record from this part of the brain. Here's just an example recording of 10 touches that are happening. And each of these little blips of activity that you see in electrophysiology is the field potentials that you record on the brain surface. I have them zoomed in over here because they happen quite quickly. So we touch the whisker and you see this burst of this field potential, that touch evoked activity on the cortical surface. But then at the same time, directly through that grid that is again sitting on the surface, we can image, I have 50 cells here, but we're consistently getting like 300 or 400 cells that we can record the activity of all these individual cells at the same time. And then we use this to kind of understand the how these individual neurons and the activity of individual neurons, or this population of cells, was contributing to those field potentials that you can record on the cortical surface, right? So I've worked quite a bit on this space on developing these polymer-based arrays that are quite transparent that we can image through. But again, kind of the drawback here, and to the majority of this field, not exclusively, is that whenever you're recording from the surface, that's definitely useful, but you largely can't record from individual cells using electrophysiology from the surface. You can record field potentials, you can record beautiful oscillations and activity that kind of propagating across the brain, but it's really difficult to record from individual neurons whenever you're doing something like laying this on the brain surface like what we're doing here. So again, this was all during my postdoc, and so now what we're doing in my lab at Vanderbilt, and our kind of vision is a platform that looks something more like this, where we still have those flexible and mostly transparent polymer-based devices, but now we can implant them and record across, in this case, cortex, or maybe even deeper into the brain if you want to, if you're interested in doing that. And what that allows us to do is record electrophysiology, just like what we were doing before from the surface, but in this case, we can record both local field potentials and record from single neurons electrically at the same time. So again, that's the benefit of electrophysiology, right? We want to get that high temporal resolution, single spike activity coming out of the brain. And we can do that with these implanted electrode arrays. And then we can leverage all the other aspects of this polymer-based probe to move everything out of the way, and also the transparency here to have our objective over the brain surface so we can record optically as well. Yes. Yes, perfect.
It doesn't have refractive index issues that it introduces for aligning cells that are
Sure. Yeah. So the question was, does the polymer-based material reduce the imaging quality? Maybe would you say that? Or yeah, it does, right? So it's actually not a huge problem that we found at the cellular level, right? So imaging cells, I did some comparisons. So for example, here of the probe on the brain surface versus not, there is actually not a big difference in imaging quality. But it did come into play whenever you tried to image small things. So we did some dendritic imaging as well, and that was really challenging through here because it basically changes your the resolution that you have, right? Because of things like it spreads the light and you no longer have a pinpoint. And so at the cellular scale actually was not a big problem, but when you try to image small things, it definitely is. And all these factors are very different when it comes to something like this that I'll see at the show, because you no longer have a probe that's covering this entire imaging view, but just one little sliver of it, basically. So it becomes less of a problem in this case. Yeah, great question. It really just depends on your question. I mean, I find this data more useful for doing like understanding like the neural circuit basis of behavior. But I think that, for example, a lot of the issues in terms of maybe some other issues could be like noise-induced or light-induced artifacts are minimal here. Because, so like, for example, the field of view would be like right here, right? This one sliver, you're only scanning right here. And so overall, we'll see some images later, but overall, you're spending very little time imaging through the probe itself, right? And we'll see some more examples of that here in a few slides. So here's kind of the vision. We're not quite there yet. We're getting really close. Again, we've only been open for about a year. And so the devices that we make in my lab at Vanderbilt, they look like this. This is, again, a very flexible probe. We've manufactured all of these in-house at Vanderbilt in microfabrication facilities. And so we can fabricate 10 to 15 probes. We call these a wafer. It's about a 4-inch disc that you can hold in your hand. And each of these is an individual probe made out of the polymer. Here's a different design. These are just the only difference that these are longer. These are made for, in this case, monkeys, a non-human primate collaborator. Everybody in my lab learns the first thing to do is learn how to fabricate these probes. So it's a multi-step process using very well-developed clean room-based techniques to fabricate these. Happy to talk about it more. We're really not doing anything too crazy or overly innovative on the front of actually manufacturing these. It's a very well-developed process at this point. And then whenever we want to use these, what we do is we submerge the whole thing in water, we take tweezers, and this will just peel up from this really hard substrate. And so the majority of this probe is basically just cabling. Really, the only thing that does any work is just this very 1 millimeter portion that actually goes into the brain. That looks something like this. So we have, in this case, a 1 millimeter long electrode array. In this case, there's 32 channels where we definitely want to scale up in the coming months and years. But right now, we're just trying to get things working. So 32 channel electrode array, cellular scale electrodes, about a 25 micron diameter electrode array. You need these to be small if you want to interface with and record from individual neurons. And then also the width is around 300 microns. The thickness, though, is incredibly thin. So about 5 or 6 microns thick. So, 1/20th the diameter of a hair. And that's what is really the main part that makes this device flexible. Also, this is something made out of parylene, if you're familiar with parylene. That's actual polymer. It's very common in like medical implants to encapsulate electronics. And we basically use that material, but as the substrate for our electronic device. Over here, you're seeing just an image of the probe. So here's the electrode array here. Here's some of the initial cabling. Overall, even though this is made out of platinum, which is obviously, this black part is platinum, it's metal, it's metallic, it's not transparent. Overall, the majority of the device is quite transparent, even by itself. And we're also working on ways on making them even more transparent. So maybe you could see in this image, I pointed at this one here, which is the platinum with the polymer. Over here is a fully transparent device that's made out of the polymer plus an electrode material that is also completely transparent. And so if we zoom in on that a little bit more, you can see here's a fully transparent neural probe. So it still has the polymer. Here's side by side the platinum with something that we call indium tin oxide. It's called ITO. And so this also has electrodes and electrode arrays, but the actual conductor is made out of something called ITO, which is also very transparent. And so this, side by side. So this would be much more transparent, more optically compatible. Full disclosure, these don't work that well yet. They're like, it's a conductor, but it's like not as good of a conductor as platinum. And so we're still working out the kinks to get this working. I'm not fully convinced that you need something ultra super transparent to do the stuff that we're going to talk about here in a few slides, but I think it's a cool project to kind of explore these unique materials for making super transparent and super optically compatible neural probes. Yeah. There's two things. Number one, this material we think is, the question was, why is it not as good? It's quite brittle, and so so whenever it flexes, you just tend to have a lot of these channels that don't work because we think it cracks. And the platinum is really robust to even like folding and even like putting a knot in your electrode. And then it's not as good of a conductor, so you can easily record field potentials, but not single unit activity. And we really want single units is really all of our projects that revolve around that. These are great questions. So how we actually connect to these things, it looks something like this. So the flexible part is down here. We've connected it to a circuit board right here. Here's just an off-the-shelf amplifier that we use. And then you're also probably wondering, how do you get a super flexible probe that's basically like spaghetti? How do you insert it into the body or into the brain? Right, there's a big mismatch there. And so we have a microwire right here that it's slotted into this slot in the back of this chip. And then the probe is here. You can see it's sort of resting on it. And these are actually physically glued together, like it's represented over here. You can see the probe is a kind of shiny, flexible part. And then that black microwire going right through the middle is just a piece of tungsten. And we glue them together with something called PEG, polyethylene glycol. It's a hydrogel that's water soluble. And so these are temporarily adhered together. And so we can basically just use this as a normal probe at this point. And so we can use the microwire to put it on a micromanipulator and guide it up to the brain and the point we want to insert, and then insert. And then over time, as we insert, as we flush that area with saline, the hydrogel will dissolve and they separate, and then we can retract the tungsten microwire and leave the probe into the brain. And so here's the actual insertion process, and here's what it looks like. But picture kind of showed up a little bit strange, but I'm not sure why it's pixelated like that. But here's the probe kind of fully inserted. The microwire is still there. We take it out in just a minute. And this also highlights the flexibility, so you can kind of fold the probe over off to the side once it's inserted into the brain. All right, and then again, we retract this out using another micromanipulator. All right, so this is how we actually get these flexible probes into the brain. We just got our first recording maybe like 6 months ago, right? So we spent almost a year kind of developing the probe, another several months getting good at the surgeries and at this implantation procedure. In like December or so, we started actually doing recordings, and so we've kind of been trying to optimize, how do we get single units more consistently? It's largely due to the insertion speed we're finding. And so here's just some spontaneous activity, in this case from a mouse brain. So just a sliver of electrophysiology and then some single unit activity that we've sorted from this data. And then we're also doing sensory evoked activity as well. So in this animal, this was all done in visual cortex. And so you can really easily implant in visual cortex and then deliver visual stimuli to the animal. And these animals are awake for these recordings. They're head fixed, but they're awake. And here's some local field potential, like visually evoked potentials that you can record across the cortical depth. So these are all inserted about 1 millimeter deep. So we're recording it across cortical layers in the mouse brain in this case. And so that's a good intro to these flexible devices. We'll talk a little bit more about imaging here in just a few slides, but I do want to, while we're on the subject of strictly electrophysiology, I want to talk about how we're also translating some of these to non-human primate recordings as well. And so why would you want to take a flexible device, go through the trouble of these ultra flexible devices for a larger brain? And this next slide is a little bit bloody, I would say. It's not work from my lab. Just if you're, I pulled it up before the talk. So if just bear with me a few minutes if you're not into that kind of stuff. But here's this is in this case, I tried to find one for a non-human primate. I couldn't find this, but this is a silicon probe recording from that NeuroPixels probe that we talked about at the beginning of the talk in a human brain in this case. And so this is a video. And so this probe has been inserted. You can just barely, that little black sliver right there is the silicon probe actually going in. This is a craniotomy, and you can see the huge amount of movement that's happening, right? And so this is about a plus, or this can be up to like a millimeter of movement in either direction. It tends to be an average about like half a millimeter. But there's just even in the mouse brain, we see this, it's just not as big of a problem in a mouse brain. But in larger animals like monkeys and also humans, the brain has a huge amount of movement. It can jostle up and down. And so again, you have this really stiff fork into the brain, and you have the brain up against it, and it's just going up and down. It's just shearing constantly. You're trying to record from a tiny 10 microns diameter cell, and that cell is just bumping up and down your entire electrode. And so these recordings can be very challenging because of the movement that's involved. And so, well, we don't really have any evidence of this, but the hope is that, if these devices that we do in a mouse brain, if they're very flexible, rather than the brain kind of shearing up and down against this really stiff rigid probe, that our flexible probes will kind of bounce with the brain and move with the brain, right? We have a little bit of evidence that this is the case. We do this in like brain phantom, these gel recordings. We haven't shown that's definitive in an actual animal, but we think that would be the case. So overall, a flexible probe could give you just a more stable recording, both in an acute setting and also in a long-term chronic implant as well. So there's certain challenges, as you can imagine, with translating to a large animal, right? So we can do this quite comfortably in a mouse at this point, and we're just barely getting this to work in a larger animal. The number one challenge is the dura. And so the dura is the enemy of our lab, we say. So it's this little membrane on top of the brain. All animals have it. Here's just a good representation of the dura has been sliced right here. So it kind of gives you there's the exposed brain, and then that little membrane is still here. So in all of our recordings, we have to like either slice or remove the dura in a mouse. And in a non-human primate, these becomes more of an issue because the dura can be really, really thick, and it can regrow quite quickly. And so the dura is just always there. It's really difficult to penetrate through it. And so what people normally do if they do a neurophysiology recording in a monkey, for example, what they will do is they call it a guide tube. It's just a syringe, basically. It's like a tiny syringe that's sharp. And you can see here that they put the guide tube in first because it's very rigid and very robust, and it can penetrate through that dura layer that's right here. And then once they penetrate using the guide tube, they then bring in their neural probe, and they can slide it through the center of that guide tube and easily penetrate into the brain tissue. And so this is what our collaborators and many non-human primate labs do currently for these recordings. And so we just wanted to adapt that process. We didn't want to get them a new fancy insertion technique. We wanted to take our flexible probes and let them have their exact same setup, their exact same micromanipulators, their exact same guide tube approach, and adapt it for our flexible probes. And so here's this work here. Really, from the engineering standpoint, all we had to do is make the probes longer because of the larger animal and kind of accommodate the surgical platform for our collaborators. And what they do is they implant this black canister here is it's just basically a chronic window to the brain. The animal has it for years at a time for access to the brain. And then we have our guide tube here that kind of penetrates into the brain first. And then we have it's a 3D printed apparatus that kind of attaches to a micromanipulator for inserting this probe into the monkey brain. And so I'm trying to keep it cool, but this worked just like 2 weeks ago for the first time. We're trying to, working with it for months at a time, redesigning the probes, adapting and getting the actual surgical process working. We just got our first recording just a couple of weeks ago. And so this is really promising. So we only got a couple of spikes in this case, a couple of single unit activity, but really promising that in the next few months, we'll be able to consistently get these recordings going in a non-human primate. This animal is also awake. So there have been some flexible electro recordings done in monkeys in anesthetized animals. Only very, very few people have done it in a wake setting. And so this animal has also been trained to do this working memory task, which is working memory is very much facilitated by the prefrontal cortex. And so our idea here is to eventually get to very long-term implants that stay in prefrontal cortex for months at a time as the animal is performing these different tasks, right? And so that was, again, just somewhat of a segue into how we are translating these devices to larger animals. And now we're going to kind of go back to one of our core technological focus is to combine these devices with imaging so we can get this very high dimensional holistic data from the brain as these animals are performing tasks and learning new behaviors, right? And so again, the reason that this is possible is because we can move everything off to the side for an objective and also image through these polymers as well. They're transparent. And so what this looks like in this case just a gel. This is an agar gel, kind of meant to simulate brain tissue, and the gel's been fitted with, we put fluorescent beads in the gel. And I like this image because you can see how our flexible probe, this is the surface of the gel. You can see how the probe bends and goes into the gel. The probe physically goes down to about 800 microns, but we just can't visualize it. This kind of goes back to these light artifacts and everything. It's so thin, right? You're just imaging a tiny 5-micron cross-section that's not fluorescent. You can't really visualize the probe much deeper than like 300 microns or so, which is right about this threshold. These fluorescent beads are quite bright, so we can image them very deep in this tissue. This is just a good representation of a flexible probe kind of going in. And again, this is just a Z stack, but in a live imaging experiment, we would choose one Z plane and kind of constantly scan across that one Z plane that you see. And this is the exact same image, but just this is a 3D reconstruction of this image. And so this is what it looks like. Again, just a good representation of what the experiment would look like in a brain. And we're just now getting this working in a live animal as well. So in this surgery that I showed you before, this is the exact same prep, and just a few steps later, after we retracted this microwire, we then capped that entire craniotomy with a piece of cover glass. That's what people do to do normal chronic imaging in a mouse. And so it's kind of hard to see, but this cover glass is kind of pressing down on the brain. It's pressing the electrode up against the brain, also kind of holding the electrodes all in place. And now we can take our animal and move it, again, head fixed under a 2-photon microscope and image this area as well. And so here's what those images look like. So again, we're imaging about a 1 millimeter square area around the probe. Over here in this video that's about to play, you're going to see a shadow over here, and that's from the probe itself that's on the surface. And here's the video. So this is, what I would consider to be on par with the imaging quality that you would get from a normal 2-photon imaging experiment, even in the absence of a probe there. And so again, the probe is kind of laying on the surface of the brain right here. It's actually inserted right about here, and so it's going vertically into the brain right at this point. Just barely, if you're really close, you can see this circle and this one here are actually electrodes that you can see. And again, also just for a little bit more detail, we're imaging GCamp activity, if you're familiar with that. So it's a calcium indicator. So as these cells fluoresce and as they're active, they become brighter. So you can, again, we haven't been able to, this data is so new that we actually haven't developed the pipeline yet to actually extract the fluorescence traces, but there's probably something around 400 cells within this field of view. So we did this surgery about a month ago or so. And so here's the recording on day 1, whenever we actually did the implant. Over here is day 24. Again, the saturation on this projector is a little bit strange, but we're moving towards not just doing this in an acute setting, but doing chronic electrophysiology and chronic imaging across hopefully 1 to 2 months, hopefully even longer than that in these animals. Again, the goal here would be to do these recordings as the animals are learning certain tasks, as they're learning that lever press task or learning other type of behaviors, to get both electrophysiological data and optical data from the brain at the same time. And again, we're also interested in doing this across scales too, right? And so in this 1 month data or so, we can also zoom in. Here's the shadow of the probe right here again. And this video over here is zoomed in on this one little section, and we're not limited to imaging cellular activity. In this case, this is excitatory neurons. We're also really confident we can zoom in and image subcellular dynamics, image dendrites, image hopefully dendritic spines, image axonal inputs to this area as well, right? So really high quality data across these different modalities and across different spatial scales. So that's great timing. This is the first seminar I've given as faculty, and so I was a little bit worried about time and how long this was going to take. But hopefully that gives you a good sense of kind of everything that my lab is working on: neurotechnologies, flexible electrodes, polymer-based devices, integrating those with optical imaging to get multimodal data from the brain, but also translating these devices to large animals and non-human primates as well. So big thanks to my lab. I've really been lucky to get a really wonderful group of students that have done really amazing work, and also thanks to all of our collaborators and funding as well. So I'll be happy to take any questions. Thank you so much. Yeah, sure.
Individual neurons and that are next to an electrode, I couldn't see it at all.
I don't know that we can. So we can image, let me go to maybe this video. So images, can we see individual neurons right next to an electrode? I would love to be able to do this. So you can see, like here, again, it looks better on my screen, but you can see fluorescence and individual cells right here. We'd love to be able to like record, okay, which electrode is that? And can we correlate the electrophysiology with that cell or a couple of cells right there? We'd love to be able to do that. I don't know if feasibly how well we can do that, but yeah, in theory, we could maybe do something similar.
Well, then the question becomes, and I don't mean to
Sure. I see where it's going. Yeah, sure.
What's the point? Because
Sure.
you've got two independent methods.
Sure. So a few different ones. So one, this is the electrophysiology would be a laminar recording, right? And so you're recording not just so this imaging would be happening at a single plane, right? But our probe can record across depth, and we're also limited in how deep we can image. When the probe, we're inserting at 1 millimeter, we could go really deep, right? To see how a deep brain region is interacting with a cortical region, for example, right? We just have a lot of flexibility there. And we can also image axons and other things too.
But
right?
you could do that independent of the photon, of the...
Sure, I think you could. But I think this maybe another example would be doing electrophysiology while also monitoring something like axonal inputs to this area, right? So like we know that like higher order feedback is really critical for learning, but you can't record from axons with electrophysiology, or it's really difficult to know, what is coming from axons. So you could record, so instead of seeing cells, you could be recording axonal inputs from prefrontal cortex or some deep brain region and seeing, how those are changing over the course of learning while you're also capturing cortical activity with electrophysiology. So yeah, so I think the cellular imaging with electrophysiology is maybe the easiest to demonstrate, but not the best application for study, right? It's more like the subcellular stuff, or inhibitory cells are also really difficult to record from electrically.
I wonder if this may be, I don't know if you have all the pieces working, but when you have your flexible electrode that's orthogonal to your imaging plane, I wonder if that's a system that can allow you to test some of the models of cortical processing using, say, artificial intelligence, where you would be getting recordings simultaneously from a column, say, so you could be in a visual or in a somatosensory area, and obviously it wouldn't be useful from a superficial layer, but if you can get down into a fourth or fifth layer in the cortex and actually sort of see what the ensemble output is of hypothesized.
Sure, I like maybe wording it slightly differently. I'd be really interested in like how that, we think, the microcircuit along a column is really critical for cortical computations, and how that's interacting with the broader population.
With the breadth,
yeah, that's like right up our alley in terms of the type of proposals that I actually write are actually less on the technology and more on, the next steps. Once this is working, what are we actually using it for to understand cortical computations, for example.
Yeah, thanks.
Hey, Daniel, thank you.
Thanks.
That was beautiful. As a site for Neuralink, I would be remiss not to ask you something. Do you understand the overlap in the methods of your implant? Okay.
Oh, sure.
So they're using similar setups. And if you know with their first implant, Nolan, that there were some thread regressions.
They slid out. Yeah.
Okay.
Yeah.
And have you seen the change in the implantation depth due to that brain movement you showed? So independent of the transparency thing here, I guess my question is more about the flexibility of these electrodes and if you see any of that translating to like current therapeutics in people.
Yeah. So I think, Neuralink's been a pain in my ass because they're doing so well, right? They're like, they're doing such amazing, just light years away from where we even are, right? But I think, maybe rephrase what was the actual question?
On the flexibility properties of the electrodes.
Yeah.
And the amount of brain movement that we see in people compared to maybe the small animal models.
Sure. So we actually saw, again, we've only done this like one time in a mouse. This probe seems to actually have sync for some reason. Like in all my proposals, for example, we talk about mitigating the probe slipping out. For some reason, ours went deeper. I don't know why. But that definitely, for all of our chronic, like non-human primate studies, definitely what we worry about more so is the probe slowly slipping out. And so I think one reason that it happened here is that the way we capped it, maybe, I don't know what the difference is between this versus completely sealing it up, with like dental acrylic and stuff like that. I can't tell you why ours seem to have consistently sink instead of retracting. But I think it's a really interesting problem to kind of how do we keep these anchored wherever you put them, right? Some type of geometry in the probe, some type of like little barbs or something that causes more damage though, to barbs to kind of hold it in place. But yeah, I think definitely in our, I'm being more and more convinced that probes slipping out are not going to be a problem for our small animal studies, but for our large animals, definitely, right? I think that's the number one thing that we think about, how do we actually hold these in place, right? And so I'm not sure what Neuralink is. So Neuralink is just implanting deeper now?
They public domain, they've said 8 mm depth instead of 4.
8. Are they even in cortex at that point still?
Right, exactly. So that's part of the question. But then they have the columnar organization, so some of the channels are in the area they want. But I think, the main thing with your micromanufacturing expertise is like that barb or whatever anchoring mechanism, that could be very, very useful.
I've been dying for an undergrad to take this on, like kind of make a brain phantom that allows us to simulate movement and simulate, like image how much the probe is moving and then testing different geometries for holding it in place,
Well, I'll be watching closely.
Awesome, thanks.
Hi. Have you thought about doing ICMS or deep brain stimulation to look at simultaneous imaging at the cortex to see?
Interesting. So stimulate with the probes basically?
Stimulating doing ICMS, because when you do ICMS, the mechanisms of how and what neurons you're activating, that's not well understood. So that could be a good problem where you could use your tool to stimulate the neurons and look at the response profile.
There's actually one group that's definitely ahead of it. So when it comes to, I showed these slides about kind of imaging. Let me pull it up really quick. There are groups out there doing surface recordings and imaging. There's only one other paper of a geometry similar to ours that's been inserted. And then they also image, and they did that actually. The group's her name's Lan Luan. She's at Rice University, and they didn't record with their device. They did stimulation and then imaged cells that were activated in like the increasing current activated more cells at a more diameter, things like that. So you should definitely look at it. It's a really interesting problem, and they want to basically understand electrical stimulation at a more fundamental level to develop better.
Also DBS. Imaging the cortex during DBS.
And deep brain. So our monkey collaborator, he does deep brain stimulation. So that's like our long-term goal, is to incorporate both flexible deep brain stimulation that's chronic and more biocompatible, all these different things.
Thanks.
Other questions? How long do your arrays last? You went to 24 days, but do they?
I don't know. Ask me.
It's too early to say.
Ask me in a year. I think so. I mean, full disclosure for this little bit of data that I did show, this probe in this case for this data was actually non-functional. So we actually didn't record in this case because it's easier to insert a probe that's not connected to all its connectors. So we're just wanted to see, can we actually image? Can we image chronically? All these different things. Electrically, I mean, there's no reason. I'd be really happy. I think a month is going to be easy. I think 2 months a little bit harder. I think if you want to go out, it's whenever you start going out to like the year mark that things start to, the polymer itself can break down, things, the saline starts to kind of leak in. That's whenever you start to have a lot of problems. For the time scales that we're very much interested in, 3 months, 6 months or something, I think is very, very achievable to do that long term.
Other questions?
This might be a little crazy, but is it, if you look at this, the imaging shows over a wide area, X, Y, and the Z is not so deep.
Yeah.
Is it possible to do an optical probe placing some kind of fibers or something there and have it be able to go in a vertical position and then record from neurons that are firing that may be further away than you can actually get from a contact?
Yes, so maybe instead of delivering the excitation from here, we deliver the excitation from like a photonic probe deep, for example.
Yes.
Yeah, I think it's possible. I don't think it'd be possible to do it with 2 photons. Everything we're doing here is multi-photon, but you could definitely incorporate like a light guide that spits out blue light here, so it'd be
Right.
single photon. So I don't know in that case how useful. Maybe you could still get it to work, but you can definitely use single photon. I don't think anybody's done like 2 photon light delivery through a light guide. But yeah, or another one could just be optogenetic stimulation too, right? Instead of imaging, actually stimulating the light as well deep. We could also do that.
Right.
Interesting.
Okay. Any other questions? If not, thank you.
Great. Thanks, everybody. Appreciate it.